Can start?
Okay, sure. Go ahead.
Yeah. So I take great pleasure in welcoming Dr. Mao Kiah from Nanyang Technological University, Singapore. So thank you, Dr. Hanmao, for accepting our invitation to speak in HIVIT 2020. This is the fourth talk in the series. So I can just read out a brief bio of yours and then start.
Okay.
Yeah. So Dr. Hanmao Kiah received his PhD degree in mathematics from Nanyang Technological University, NTU, Singapore in 2014. From 2014 to 2015, he was a Postdoctoral Research Associate at the Coordinated Science Laboratory, University of Illinois at Urbana-Champaign. From 2015 to 2018, he was a lecturer at the School of Physical and Mathematical Sciences, NTU Singapore. Currently, he is an assistant professor at SPMS, NTU Singapore. His research interests include DNA-based data storage, coding theory, enumerative combinatorics, and combinatorial. So, thank you so much for accepting our invitations. Over to you.
Thank you. All right. So okay. All right. Good evening, everyone. Okay. Hope everyone is staying safe and healthy during these unusual times. Well, these times are certainly unusual. Well, it has presented this unusual opportunity for me to share some of my works in these unusual settings. All right, so I must first thank Prasad for this very kind invitation, and more generally, the entire community, the committee for conceiving this idea and putting this unusual but great workshop together. Okay, so my name is Han Mao, as Prasad said, and I'm from Nanyang Technological University. And the title of my talk here is Balancing a la Knuth for DNA-based data storage. Well, this talk is a amalgamation of a number of joint work with a number of my collaborators, and I will give them a shout out later on in my talk. Okay, so as the title implies, we'll be looking at problems that arise from DNA-based data storage and use a special technique invented by Knuth to handle them. All right, so yes, over here on the top right is Knuth, and you might know him for many things. Okay, one of them is LaTeX, another one is this series of textbooks, or rather encyclopedia. Okay, the art of computer programming, which is reading right here over here. But for those of us who work on constraint coding, he's very famous for this balancing technique. So in this talk, we're going to look at the balancing technique and adapt it to a variety of problems from DNA-based data storage. All right, so what is DNA-based data storage? Is it working? Oh, yes, it works. Okay, so DNA-based data storage means that what we'll do is to use artificial DNA strands to store our data. So instead of using 0 and 1s, we'll use the DNA bases A, T, C, and G to represent our data. Well, why DNA then? So here are some typical arguments for using DNA. But I think the best examples are around us. We are the examples. Nature has been using DNA in us to store our genetic information. So natural question is, why not use DNA to store digital files like our photos and movies? Well, to answer this question, we have to ask ourselves, can we create DNA? And can we read DNA? Well, in the last millennium, this is either not possible or very expensive. So until very recently, in 2012 and 2013, the biologists have developed the necessary technology to do this. In particular, we have these two research groups, okay, which actually try to store digital files onto artificial, or what we call synthetic DNA strands, and manage to read them back in real life. Since then, we have many more experiments that conducted. Since then, there were many more other experiments that was conducted using other technologies. Okay, so in this experiment, okay, my former colleagues, okay, at Urbana-Champaign used this thumb drive-like device to read DNA data or rather to store DNA data and then read back, okay, on this DNA thumb drive. Oh, sorry, this thumb drive-like device. Okay, and in another experiment, the group at Harvard University stored this GIF file onto some live bacteria, okay, and again managed to read back the data files. Well, we also have big companies like Microsoft who are buying into this idea. So in particular, Microsoft actually bought millions of strands of DNA in, I think, 2016 and conducted their own experiments. So what I'm talking about here is not science fiction. And to date, Microsoft holds a record for storing the largest data, which is around 200 megabytes. Okay, so okay, all right. So let me give you a high-level description of a DNA-based data storage architecture. Okay, so basically what we have is a bunch of 0 and 1s, okay, some digital data. And what we will do is to convert them into DNA codewords and use a machine called a synthesis platform to create DNA strands. After that, we will store them in some storage media. So typically what we put them is a DNA pool, which is usually a test tube. Then when we want to read them in the future, okay, we'll take them out, okay, and pass them through what I call a sequencer and invert our encoding process to retrieve the data. Of course, during this process, there might be errors like over here. And as coding theorists, our task is to design encoders and decoders so that the input data is similar to the retrieved data. Now, for those of us who work in data storage, okay, we'll realize that this diagram is same for any data storage device. Okay, what I've done here is essentially to change the nomenclature. However, there are very significant differences between the DNA devices from traditional media or devices. Okay, this then means that our traditional approaches towards coding must change. And so, in other words, okay, as a coding theorist, we have more problems to work on. And to illustrate this point, okay, here's a sample of some papers from this year's ISIT. Okay, so ISIT is the International Symposium on Information Theory. And you can see here, we have a number of papers that is on DNA-based data storage. And you also see that each paper here addresses one or more aspects of DNA storage. So again, let me emphasize that. This is only from this year's ISIT. Okay, there are many other papers from other years, earlier ISITs, and also many other brilliant results from other journals and other conferences, which I cannot fit onto this slide over here. Now, not only can I not fit all the results in one slide, I also cannot fit everything into one talk. So for today, we'll focus on one aspect, or rather a few papers that center around this notion of constraint coding plus. Okay, so specifically, we're going to look at this constraint called the Gc Balance Constraint and equip it with other capabilities like random access and error correction. So what does this Gc balance mean? Now, DNA strands are made of four nucleotide bases, okay, A, T, C, and G. And not just this, okay? The Watson-Crick pairing states that DNA sequences occur in pairs. So A is always bonded with T, and C is always bonded with G. And moreover, the bond between A and T is very different from the bond between G and C. Hence, we see that many chemical properties depend on the number of G and C in the sequence itself. So formally, we're going to call this proportion the GC content. And the word is GC balance if 50% of the bases are G or C. Okay, so DNA strings which have high GC content, which is GC rich, or low GC content, which is GC poor, are prone to both synthesis and sequencing errors. So therefore, okay, our code design is to make use of code words that are either GC balanced or almost GC balanced. So GC content close to 50%. This is Okay, so let's look at the corresponding notion for binary words. Okay, so for binary word, we can call its imbalance to refer to the absolute difference between number of ones and zeros. So a binary word is balanced if its imbalance is at most one. Okay, so in other words, the weight is close to n/2. So in this example here, 0, 0, 1, 1, 1, 0, 0, 1, we have 4 zeros and 4 ones. So therefore the imbalance is 0, and so we say that this binary word is balanced. Okay, so what's the relationship between balanced binary words and Gc balance? Well, we have this simple correspondence over here. Okay, to understand this, we require a mapping between A, T, C, and G to 2-bit pairs. So given a DNA sequence, okay, what I'm going to do is to have a correspondence to map this into 2 binary sequences and go place them on top of each other. Okay, so basically A maps to 0, 0, T maps to 0, 1, C to 1, 0, and G to 1, 1. Okay, and with this correspondence, you can check that the GC content is determined entirely by the weight of the upper sequence. So in this example, the upper sequence is of weight 4, and therefore the GC content of the corresponding sequence, okay, is just 4/8, which is 50%. So in other words, okay, a DNA sequence is GC balanced if and only if its upper sequence is balanced. Okay, and a low imbalance of the upper binary sequence also implies that the GC content of the corresponding DNA sequence is close to 50%. And so for this reason, okay, in some parts of my talk, I will simply look at just binary balanced words and mention in passing that the methods can extend to DNA alphabet. Okay, so let's introduce the central technique of this talk, Knuth's balancing technique. Well, this is a technique that encodes binary messages of length n into binary balanced words in linear time using log n plus a little bit of redundant bits. Now, before I go and describe this method, okay, we're going to note that the balancing technique is almost optimal in the following sense. Okay, the calculations over here, okay, shows that the number of balanced words encodes around n minus half log n bits of information. So half log n bits of redundancy is always necessary. And here we will use only log n redundant bits. So in some sense, okay, we are slightly off. Now, the efficiency in redundancy is one aspect of the technique. Okay, to me personally, it is the simplicity and its efficiency of the encoding process that is the beauty of the method. Okay, so let me explain how the method works with a simple example. So here we have a binary message of length 10, and we want to transform it to a balanced word. Okay, what should we do? Well, Knuth's technique says that let's try to flip the bits one by one. When we flip the first bit, okay, this word of weight 3 now turns to weight 2, which is certainly not balanced. Then what Knuth says is, let's continue. So we flip the next bit and get weight 3. Not balanced yet, so we flip again. Get weight 4, and it seems like we are getting closer, and indeed. Okay, in the next instance, when we flip the next bit, you get a weight 5 word, which is indeed balanced. And so what we will do is to use this as our code word. However, we need to tell the receiver, okay, how many bits have been flipped. So what we need is a lookup table over here, which contains of all balanced words. So the index 4 corresponds to this balanced code word. So we append, or rather we put it as a suffix, and this will be my balanced code word. Okay, so in this case, we introduce these 5 redundant bits. Okay. And so in this talk, we will adapt Knuth's balancing technique to achieve the following objectives. Okay. So first one is balanced code with random access capabilities. Next is balanced codes that correct single nucleotide edits. And we also try to combine the balancing technique with polar codes. Okay. Now in this talk, not only are we modifying Knuth's balancing technique, we're also trying to keep to Knuth's spirit. Okay. So specifically, what we are after are explicit codes, and we are not looking at existence results. Okay, so existence results that arise from GV type arguments. Okay, also we are not looking at random constructions. And in all cases, we will provide efficient encoders and decoders. And of course, okay, we want to encode as much information as possible. So we are looking at codes with low redundancy or high rates. Okay, so this talk is divided into it's divided into 3 parts, each part focusing on enforcing the GC balance constraint while addressing another aspect, other aspects of DNA storage. And in each part, okay, I had the joy of working with my close collaborators that include students and postdocs. So in this part, we will look at random access capabilities. And here, over here, on the top left, is Hossein, who was previously a PhD student at Urbana-Champaign, and he led the first work in providing the coding design considerations. Okay, Hossein is currently at a startup, okay, in the Bay Area. Mao Kiah is here, okay, he was a postdoc with me at NTU. The balancing technique that you will see later was introduced by him. Okay, and Mao Kiah is currently at Ben Gurion in Israel. Okay, so why random access? Well, this is due to the unordered nature of DNA strands. So unlike traditional media, where we store information on DNA strands, the strands do not keep to its place. They are like fishes swimming in a bowl, and it's not clear how we can fish out a specific information block that we want to read. So my colleagues at Urbana-Champaign propose the following strategy. Okay, so the strategy is this: is to prepend these information blocks with address sequences, which we will call primers. And to retrieve the data, we will make use of what we call a reverse complement, so that via this chemical process called DNA hybridization, okay, the correct strand will be attached itself to the reverse complement, okay, and so sometimes being fished out. All right, so let me attach some pictures to the names. So again, over on the left here is Hossein, and on the right here is Ogitsa. Well, of course, in such proposals, things do not always work out perfectly. Okay, and so we need some coding strategies to minimize probability of errors. So in this part of the talk, okay, we are going to look at some design considerations for the primer sequences. Okay, so in some sense, we want these primers to be distinguishable so that we can pick out the correct strands. Now, essentially, together with Ogitsa, Hossein, and Ryan, we provided a set of design criteria for these sequences, and I'll be describing this later on. Now, before I provide the formal definitions, I'd like to highlight that the group at Urbana-Champaign, specifically Hossein and Ogitsa, actually created DNA strands based on this strategy and performed the necessary experiments to verify the feasibility of this method. So again, what we are designing are the primer sequences, which are the prefixes of our DNA strands.
Hanmo?
Yes?
This is Shashank here. I have a question. Is that a good time to pause?
Sorry, what?
Yeah, can I ask a question?
Sure. Go ahead.
Yeah, please. So, so is the primer sequence expected to be a function of the information block?
No. So, but the information block will be encoded based on the primer sequences also. Okay, and this part of the encoding, I won't be describing it for this talk. Yeah, we have a separate work on this, on how to encode the information blocks based on these primer sequences.
Okay.
Do I answer your question?
Perfect. Yeah.
Okay, cool. Okay, good. Yeah, feel free to interrupt me. Yeah. Okay, so, yeah, good. At least I know I'm talking to someone. All right. Okay. This feels much more comfortable. Okay, so, all right. So we have 4 conditions over here. And the first condition is kind of clear. Okay. The first condition is that we want our primers to belong to a block code of a certain length and a certain minimum distance. Okay. This will allow us to correct the usual substitution errors. Now, the second condition is slightly unusual, is this notion of mutually uncorrelatedness. It's characterized by this parameter kappa. Okay, so a code is kappa mutually uncorrelated if no proper prefix of length L, which is at least kappa, appears as a suffix of another code word. So in this set of sequences, okay, here we have chosen kappa to be 5. Here, so this CTGCG, okay, the prefix does not appear as any suffix over here. Okay, so if you think about it, this will actually measure how we will prevent fishing of the wrong suffixes, okay, in some sense. Okay, anyway. Okay, in the third condition is parameterized by F and refers to avoiding primer-dimer products, okay. I will not elaborate more on this condition. I will just mention that we actually look at this condition in our full paper. And finally, this is the last condition that we are after. That is, we want all my words to be Gc balanced. Well, in our first work in 2018, okay, we actually proposed iterative construction to satisfy all four design criteria that I gave earlier. Okay, in particular, we provided a iterative construction. Okay, and this, however, this construction actually requires what we call short balance primer codes. So this is not entirely explicit, and we point to some shortcomings later on. In this part of the talk, okay, instead I'm going to tell you of a more explicit construction that uses only cyclic codes. However, in my exposition, okay, we only try to satisfy 3 out of the 4 properties that I mentioned earlier. Okay, and if you're interested, okay, you can read the full paper in Transactions IT. Okay, so in fact, our technique is much more general and allows one to construct balanced codes with error correcting capabilities. So let's try to review some of the current techniques that's out there. Okay, so one of them is by Weber et al. In 2012. So this actually mimics the quite closely Knuth's balancing technique. Now, at the beginning, instead of picking any message, okay, what Weber et al. Does is to pick words from a error correcting code. So here he picks words from a block code of distance 3. And what they then do, okay, is to follow what Knuth did to flip some of the prefixes, so to get a balanced word. Now, as before, we need to record down how many bits was flipped. Now here, instead of picking any balanced word, okay, we have to be careful, okay? To maintain the minimum distance property, we need to pick out the index from another shorter balanced word. So here, this is a balance. So here, our words are of length 7, and you can see that they actually have distance 3. This will then ensure that whatever the code word that it encodes have a certain distance property. Okay, as you can see here, this construction is not entirely explicit, and it's not clear how we can efficiently construct this short balance code. And furthermore, the number of additional redundant bits that we have here is p. So in other words, okay, the distance is at most p, and in other words, the error correcting capability is limited. So in our method, we will try to remove this need for a short balance code, and what we will need is just a cyclic code. Also, the additional redundancy we introduce will always be log n. Okay, this is independent of the distance d. Okay, so here my catch is this. Okay, our technique only applies when we have a cyclic code. Okay, so unlike Weber's method, okay, which actually works for any linear code. Okay, so let's try to first revisit Knuth's balancing technique and understand why it fails. Okay, so here we have 2 words starting out with distance 3. And realize that after we apply the Knuth's method, okay, the distance is actually reduced to 2. And the reason is pretty obvious. Okay, we actually flip different bits in both cases. Okay, so that's why the distance is not preserved. Well, to preserve distance, then a natural way is to just flip the same bits. Okay, and so in our method, we always flip the first 5 bits. But then you might ask, if you flip the first 5 bits, how do I guarantee that the weight is correct? Okay, so like in this example, okay, I flip the first 5 bits and I obtain a word of weight 6, which is clearly not balanced. So what can we do? Well, here is where we need the condition of a cyclic code. Okay, what we will do now, instead of changing the number of bits we flip, we cyclically shift, rotate the words over here. So I shift all the words here to the left. Now, if you flip the first 5 bits, you realize that, okay, we actually obtain a word which is balanced. Okay, we obtain a word of weight 4. So this is our balanced word. And so as with Knuth's method, okay, we can show that there is always one cyclic shift that achieves balanceness. And since we always flip the first 5 bits, okay, the distance is always maintained. Okay, so here is a more concrete construction of the balanced error correcting code. Okay, so what we're going to do is to partition the cyclic code into different cyclic classes. And for each of the cyclic class, we find a cyclic shift such that flipping the first n/2 bits give a balanced word. And then we go and collect all these balanced representatives, and this will form our balanced code of the same distance d. Okay, now note that each class has at most n cowords. So therefore, okay, our code size is at least 2 to the power k over n. In other words, okay, our method only introduce log n redundant bits. And since this method works with any cyclic code, okay, we can use some famous family of cyclic codes like the BCH codes, and we can get a family of balanced error correcting codes, okay, with this redundancy. Also, our method can easily generalize to the DNA alphabet. Okay, so I won't go into detail, but can obtain GC balance codes by introducing only log n redundant bits. What else? Okay, now to enforce the kappa mutually uncorrelatedness, okay, we need to enforce more conditions on the input codes. So in addition to being cyclic, okay, we need to restrict our rates to be at most 1/4, and also need to make sure that all my code words and the code book actually contains the all one vector. Okay, then we can do a small adjustment to our technique. Okay, so in addition to flipping the first n/2 bits, we also flip the last symbol also. Okay, with some arguments, okay, we'll show that this is actually a almost GC balanced kappa kappa mutually uncorrelated codes. Okay, and essentially what I do here is a modification of a synchronization technique, okay, introduced by Tavares in his doctoral thesis in 1968. Okay, now it remains to provide an efficient way of encoding messages into distinct cyclic classes. Okay, but I'm running out of time for this part, so I will not go through in detail. Okay, the details can be found in our transactions paper, okay, over here. Okay, so I'll just quickly scroll through the slides. Okay, as you can see, we are again using another technique of Tavares, okay, to do the encoding. Okay, so let's move on to the next part of our code of this talk. Okay, so in this part, okay, we move away from coding of the primer sequences to the coding of the information blocks themselves. Okay, so in this part, we're just going to deal with single errors. And in fact, we're going to look at a very special class of errors called nucleotide edits. As with before, here we will require all our words to be GC balanced. So over here is my previous PhD student, Tang, who graduated last year. He's the main driver of this work, and is currently a postdoc with Kui Chai at SUTD in Singapore. And he's also continuing this line of research. Okay, so what's this nucleotide edit about? Okay, as mentioned earlier, okay, there have been numerous experimental implementations of DNA-based data storage since the first two in 2012 and 2013. And recently, the Microsoft team implemented an experiment involving millions of DNA strands. And as part of the experiment, okay, they actually reported the error rates. So basically, the usual errors of insertion, deletion, and substitution. Okay, later on, okay, another group, okay, led by Hacker et al., okay, also reported similar error rates. and also made this interesting observation. Okay, what they saw was that certain substitution errors, okay, or rather, if you want to think of it as crossover probabilities, are significantly higher than others. Okay, so specifically, T turning to C and A turning to G is much higher than the rest. Hence, motivated by this observation, we decided to look at a very special type of substitution error. So specifically, we define an edit error as a deletion, insertion, or substitution. Okay, this is the usual type of edits. Okay, now, but for the DNA alphabet, okay, we consider a subset of these edits. Okay, so in particular, a nucleotide edit. Okay, sorry, this is a name that we came out ourselves. Okay, refers to either a deletion, insertion, as before, or a substitution where A and T is replaced with C or G, or vice versa. And so in this so-called special types of edits, okay, the errors that were reportedly more likely to occur are classified as nucleotide edits. And here we will try to correct this so-called nucleotide edits. Okay, so specifically, okay, we'll look at correcting a single nucleotide edit and also try to combine this error correcting capability with the Gc Balance Constraint. Okay, as before, we want explicit codes with efficient encoders, and in this case, we actually have linear time encoders. Okay, so again, we have a full paper whereby we also talk about non-binary case and correcting single edits in general. Now, before I go in detail on the construction itself, okay, let me highlight what we achieve in terms of redundancy. Okay, to correct a single nucleotide edit, we use log n redundant bits. To correct a single nucleotide edit with the GC constraint, okay, we use 2 log n redundant bits. Now, the usual square bracket bound requires at least log n redundant bits. So what we have here is almost optimal. Okay, it's optimal in terms of the redundant bits. Now, for the binary case, okay, especially for single edits, the problem has already been solved way back in 1966 by Levenstein. Okay, so basically what he observed was that the codes found by Rashomon of tandem gods, okay, that corrects asymmetric errors can be used to correct a single insertion or a single deletion. Okay, so I'm going to refer to this as indel. Now, by modifying this code construction, okay, basically changing the modulo over here, okay, what Levenstein showed that that the same code can actually correct a single edit. Later on, in 1984, tandem gods actually proposed extended this generalized construction to correct single indels for the non-binary case. Okay. Unfortunately, okay, tandem gods constructions are unable to correct a single edit. And Levenstein's modification, okay, which works for the binary case, okay, does not work for the non-binary case. So what can we do for the DNA alphabet, which has 4 letters? Okay, so to do this, we revisit the mapping that we had before. Okay, where we write a DNA sequence into 2 binary sequences of length n. As before.
A quick question.
Yeah, sure.
So can you go to the previous slide?
Yep.
So this non-binary VT codes correct single indel, but they are unable to correct single edit. Is it? So that means your the nucleotide edit which you are talking about is not corrected by this. Is it?
Yes, correct.
Okay.
Yes.
So specifically the A to T and G to C. That's where it fails.
Precisely. Very good. Yes. Thanks.
Okay.
Good.
Yeah. Thanks.
Okay. Yes. So yeah. Yes. Okay. At least there's someone listening. Okay. All right. So where was I? Okay. Yeah. So in our mapping earlier, we observed that using this mapping, okay, the GC content is determined entirely by the weight of the upper sequence. So now let's try to observe, okay, what happens, okay, when we edit errors happen to our sequences. Okay, so specifically, if you think about this, okay, if I have an indel on the DNA sequence, okay, this will actually correspond to an indel in both the upper and the lower binary sequences. Okay, this is pretty obvious. However, for the substitution, okay, if we have a substitution error, okay, for the DNA sequence, okay, what we observe is that there is a substitution error, okay, in either the upper sequence or the lower sequence, or both. So it's an and or condition. So again, I want to emphasize the or, okay? So in this case, where C turns to G, okay, the substitution error only happens on the lower sequence, it does not affect the upper sequence. Okay, this turns out to be a problem, okay? But anyway, so very obvious is that we have a very simple code construction, okay, which is simply to require both upper and lower sequences, okay, to be part of a single edit correcting codes. Okay, this means that we will introduce a redundancy of 2 log n bits. Okay, again, this is off from the spherical game bound, which needs only log n bits. So our target is to aim for log n bits.
So Han, another quick question. So this mapping of A to 0, 0, that is our choice, right?
Yes.
So, because you are looking at only the substitutions like A to T and G to C, is there a possibility that you simply change these mappings so that you get these edits happening in both the top binary sequence and the bottom?
Okay. So okay. So later on I'll explain what if we just focus on a specific type of substitution that will happen. However, in general, okay, if you think about it, if I just allow A to change to any of the other three, right? It's by some I think some argument you realize that that is either or condition, okay? That must be one of them which will not be changed at all. Okay. However, for our special kind of edits, okay, we won't see this problem, and we'll try to make use of this fact. Okay.
Right. So this code, whatever you're constructing, handles any substitution?
No.
Okay.
For this case, no. Yeah.
Okay. No problem.
Yeah. So that's sort of illustrate what I'm going to say later on. Okay, which is here. Okay. So if we look at our special type of substitution, the nucleotide substitution, where you change AT to either C or G, okay, then this will actually force the upper sequence, okay, to always have a substitution error, okay, by this definition itself, okay, which is some sense happy coincidence, okay. And so this actually allows us to actually use a different code for the lower sequence, okay. So this is what we call a shifted VT code. So before I go into details, okay, so by changing the using this lower VT code, okay, this actually allows us to actually reduce the redundancy from 2 log n to log n, okay, plus this little bit of things here. Okay, good. So what is this shifted VT code? Okay, let me give a short introduction. Okay, so this shifted VT codes were actually introduced by Shenny et al. Recently in 2017, where the VT syndrome is taken over a much smaller modulo value. Okay, so instead of taking modulo over modulo n, you take it over modulo p. And here, p is usually chosen to be typically much smaller than n. Okay, so my redundancy is log p instead of log n. And the question is, why can we take a VT syndrome over a smaller modulo value? So again, we are going to real technical details. Okay, the broad approach is to make use of the certain knowledge that we gain over the to make use of certain knowledge gained from decoding our upper sequence, okay, to help me decode the lower sequence. Okay, so actually, so the main insight is this, so if we don't know where the error is, okay, if it's over a whole window of n, okay, we need a syndrome, get a VT syndrome taken over modulo 2n, okay, to correct a similar edit. However, if we could able to isolate it to a smaller window of length p, okay, then we can take the VT syndrome over a smaller modular value. So here in this case, we take over modulo 2p, and this will actually do the job, okay, provided we know we could isolate the error to here. And so, so again, I have glossed over many technical details, okay? And you can again look at my full paper, our full paper on arXiv. Okay, so for the actual code, okay, in addition to this shifted VT syndrome, okay, we also enforce what do you call it? We also enforce a constraint on the upper sequence, okay? Basically, we restrict the maximum run of the same bits to be at most p, okay? I don't want to go through the details, okay, but here is the decoding algorithm. And if you're interested, okay, look into our arXiv paper. But let me just mention how we estimate the number of redundant bits. Because, okay, in this, our construction over here, we have a choice for the parameter p. And to achieve optimal redundancy, okay, what we do is to set p to be roughly log n. And it turns out that one redundant bit is sufficient to enforce the maximum run condition. And then straightforward computations will tell you that the number of redundancy that we have now is just log n plus log n. Okay, so p being the log n. Okay, so by doing this simple trick, okay, we actually are able to correct a single nucleotide edit, okay, using only log n redundant bits. Now to enforce the GC balance, okay, we can do the following, okay. Okay, in the interest of time, okay, I'm just going to demonstrate the encoder via a simple example, okay, and yeah, you can check out the details over here. Okay, so here, I want to get GC balance, okay, and also satisfy all these conditions here. Here, n is chosen to be 16 and p to be 8. So what we are given here is a 19-bit message, and we want to encode the message into two sequences, two codeword sequences, the upper and the lower, so that these constraints are met. Okay, so the green positions are where we're going to put our data bits, and the other positions, the 13 other positions, is our redundant bits. Okay, so first we'll try to get it to be balanced. So here we just need to look at the upper sequence, and so we use Knuth's technique. Okay, so using Knuth's technique, we just go flip the first 3 bits, okay, and we get something of weight 5. Okay, and then to record how many bits we flip, we use the blue cells over here. Okay, so 0011 is a binary representation of 3. Okay, telling me that I've flipped 3 bits here. Then we fill in the yellow bits, okay, so that the syndrome, okay, is what we want, okay, which is here I'm going to set it as 0 modulo 32. Okay, why this works, okay, I don't want to go into detail, okay, but basically again, you can check out our paper over here. Okay, no, it's not too difficult. Okay, so after filling the yellow ones, we fill in the orange ones, okay, and orange ones will help us ensure that the shifted VT syndrome is correct. Okay, that this shifted VT syndrome, the shifted VT syndrome over here is equal to 0 mod 16. Okay, so using these 4 steps, okay, after using these 4 steps, okay, we do our mapping again and get back our bases A, T, C, and G. Okay, and it turns out in this example, okay, we actually obtain a GC content of 50%. Again, I've shared away certain details, okay, like how I go and enforce this run length condition over here. Okay, well, just believe that it can be done, and it isn't too difficult. Okay, so this concludes the second part of my talk, whereby we look at constructions of where we try to correct single indels and edits. Okay, in our full paper on arXiv, okay, we actually propose non-binary codes that correct a single edit. Okay, whereby this edit need not be the substitution that we have earlier. Okay, so basically all types of edits. Okay, all types of substitution. Okay, and we also still achieve log n redundancy. But to do this, we need.
Hanma, Prashant here. Yeah. So can you perhaps just give a brief idea of how do you decode the previous code?
How to decode? Oh, okay.
If there's a single insertion or deletion.
Oh, okay. So we have the okay. So if there's an insertion or deletion, right? So you simply break up into the 2 sequences over here, right? Then we follow our procedure as before. Okay, so you Let's just go back. Okay, so I'll talk about the unbalanced case. Maybe this is a bit easier. Yeah. Okay, so the upper sequences has a usual VT syndrome. Okay, so I can decode the single edit, okay, the single insertion, deletion, or substitution, right? Okay, so now the question is, what happens? Okay, so what's interesting here is that for the nucleotide edit, right, if there's no error on the first sequence, okay, it turns out that the bottom sequence must have no errors also. Okay, so that's a helpful part. Okay, otherwise we know where the error occurs on the lower sequence. Okay, and so you can just use your syndrome to check again. Okay, and then for deletion and insertion, it's pretty straightforward. Okay, yeah,
because they're just VT codes.
yeah. Actually, yeah, sort of, yeah. Okay, does this make sense?
Yeah.
Okay, cool. Okay. All right. So okay. Yeah. Okay, so next. Okay, so let's move on to our last part of our talk. Oh yes, I'm making good in time. Good. Okay, so in the previous part, right, what we did was to focus on correcting single errors. Okay, this was primarily because the error rates are low, and using current technologies, okay, we only can synthesize very short DNA strands. Okay, strands of length 100 and 200, 100 to 200. So we expect at most around one error in each codeword. Okay, but now for certain new sequencing platforms, okay, which actually does the sequencing much faster, error rates are much higher. So it's natural to ask whether we can deal with more errors. And so in this part, okay, we're going to see if we can combine Knuth's balancing technique, okay, with a beautiful class of code called the polar codes. Okay, and here we have two students. Okay, so on the left is an undergrad from IIT Delhi. On the right is Hanwen. Sorry, I didn't mention his name. Okay, this is Akash from IIT Delhi, an undergrad. And on the right is Hanwen from UC San Diego. Hanwen is a PhD student of Alex over here. And and Hanwen's primary area of research is in polar codes. This work was actually done when both Akash and Hanwen visited me in Singapore during the last summer, okay, before this pandemic. Okay, anyway, so we are trying to combine the balancing technique with polar codes. Okay, so what are polar codes? Well, so what are polar codes? Okay, all right, so I will give a more detailed definition later on, okay, but essentially polar codes were invented by Arikan back in 2000, around 10 years ago, in 2009, and they are a class of codes with low encoding and decoding complexities. Well, initially, Arikan demonstrated that these codes achieve capacity for the binary semantic channel, but within the last 10 years, okay, there have been many works extending similar results, okay, capacity achieving results for other channels. And of significance, okay, polar codes have been adapted for the insertion and deletion channels, and this turns out to be particularly useful for DNA-based data storage. However, polar codewords are generally not balanced, and so in this part, we'll find a way to ensure that the transmitted words are balanced while retaining the low complexities of the original polar and encoding and decoding algorithms. Okay, so to do so, we're going to apply this notion of linear balancing sets introduced by Mazumdar et al. In 2010. Okay, so in this work, a balancing set refers to a set of vectors such that whenever you give me a binary word or x, okay, I can look into this set of balancing vectors and pick out one, say b, and when we add b to x, the imbalance, okay, will turn out to be 0. In other words, okay, the sum x plus b is balanced. And if you put this in the context of a Knuth scheme, okay, then Knuth's balancing set is simply a set of vectors whereby each vector consists of a run of ones followed by a run of zeros. Okay. And what Mazumdar et al. Was interested in was balancing sets that were linear. That is, they form a linear subspace. So in this example, okay, so let's just be more concrete and look at the case for n equals to 4. So in Knuth's scheme, the balancing set will be just 1, 0, 0, 0, signifying the flipping the first bit, 1, 1, 0, 0, 1, 1, 0, 1, 1, 1, 1, okay, flipping all 4 bits. This is clearly not a linear space. Now, in contrast, okay, what I have here is a linear balancing set, okay, whereby it's a span of 1, 0, 0, 0, and 1, 0, 1, 0. And you can check here, okay, exhaustively that I can balance any word they give me, okay, using this linear subspace. Okay, so why are we interested in linear balancing sets? Well, what Mazumdar et al. Showed was that one can combine a linear balancing set with a linear code correct errors. Okay, so let's just illustrate this with a small example over here. So in this example, we have a 11 by 16 matrix. Okay, we actually generates a linear 16, 11, 4 code. In fact, this code over here is the extended Hamming code that corrects a single error. Now, from this 11 rows, okay, I'm going to pick out 9 of, I highlighted, how many of them? See, I highlighted some of them, okay, to be green color, okay, and you have to trust me, okay. Basically, this green rows, okay, will span a linear balancing set. And let's color the remaining ones blue, and let C prime be the span of the remaining rows. So, if you give me a word C in C prime, say this word over here, which is this row over here, okay? By definition of the balancing set, okay, I can find a B among the green rows, okay, such that when I add the two of them, I get a balanced word, okay? And this will be a balanced word that I send over. Well, there's nothing surprising, okay? That follows from definition. But what is important is this, okay? That the word C plus B, okay, belongs to original single error correcting codes. So in other words, okay, even C plus B, if there's a single error, I will be able to correct them. Okay. So also in particular, we can apply any decoding algorithm that we had for the original code C. And of course, this works for any linear codes in general. Okay. Again, let me emphasize that what we are doing here is simply to inherit the decoding algorithm of the original code C for error correcting purposes. Now, in the same work by Mazumdar et al., they actually showed that if you randomly pick a linear space of dimension 3 over 2 log n, okay, with high probability, okay, this is actually a balancing set. However, okay, verifying that B is a balancing set, okay, is computationally difficult, okay, and moreover, okay, if you randomly pick a subspace, okay, you actually have in essence of a random linear code, and decoding a random linear code is not computationally easy.
Han, a quick question.
Mm-hmm.
So this question is about Mazumdar's work.
Mm-hmm.
So what was the motivation for them to come kind of linear balancing sets? Do you know?
Yeah. So basically, is to create balancing codes with a balanced error correcting code.
Okay.
Yeah.
Okay.
So basically, like what I described earlier. Okay. To be able to take any linear code and just pick out some of them as for balancing purposes, so that the resulting code is actually still balanced and still can correct errors.
Right.
Yeah.
Okay. Thank you.
Okay. So for this part of the talk, okay, what we're going to do is to look at a more explicit set of balancing words, okay, more explicit linear balancing sets, and we're going to use the technique, okay, to balance polar codes. Okay. Okay, so what are polar codes? Okay, let me present a very, very, very simplified explanation. Okay, so polar codes are best described by this matrix GM, okay, which is also known as the polar transformation matrix. Okay, to obtain this GM, what we do is to take a tensor product of this 2 by 2 matrix, this 1, 0, 1, 1 matrix, okay? Take the tensor product to obtain a square matrix of dimensions 2 to the power m by 2 to the power m. So in this example, m here is 4, so n here is 16, okay, which is 2 to the power m. Okay, so this is a 16 by 6 matrix that represents G4. Now, I also colored certain rows of GM red and the other rows yellow. Okay, the red rows are known as the frozen rows, while the yellow rows are used to carry information. And here is how we obtain polar cowords, okay? Our cowords will essentially be linear combinations of rows of GM, where the coefficients are obtained from this column over here, okay? You see that I colored the certain cells red. Okay, so for the red rows, I color it pink, and for the yellow rows, I color it orange. Okay, so for the pink cells, okay, these values will always be fixed, and typically we fix them to be 0, as in this case. So we can actually so-called freeze the cells. And for the orange cells, they can be anything, and these orange cells are basically used to transmit information. So in this example, this polar code transmits 11 bits of information. Now, for this particular set of information bits, okay, we have a 1 here and 0 elsewhere. Okay, so this will actually correspond to this row over here. So my polar code word is this word. Okay, and here it has weight 4, which is not balanced. Now, given this setup, okay, it's not difficult to see that encoding is efficient. Basically, doing it in n log n time. However, what is nice is that, or more important, is that Arikan showed that there's this decoding method called the successive cancellation decoding, whereby you can use it to efficiently and correctly recover a message with high probability. And also, you can show that you can achieve capacity, or rather, you can achieve rates that are close to capacity. Okay, so I cannot emphasize this more than enough, okay? I have overly simplified the description of polar codes, okay? There are many details that I have omitted, like how successive cancellation decoding works, why it works, and how to choose the frozen indices so that this decoding works, okay? The details here are very technical, and one can devote months to understanding why it works. But nevertheless, okay, fortunately for us, what we need is really just the encoding, and the encoding part is a bit easier to understand. So let us see if we can have a way to make sure that the code words transmitted are balanced or almost balanced. So here we want to transform this to a word which is close to weight 8. Okay, so again, I'm going to set aside some of the information indices for balancing purposes. So here I set aside these few green rows. Okay, so now we only transmit only on the if we really can have the freedom to set bits on the orange cells. Okay, so let's say we want to transmit this information 11000. Okay, so we fill up the information bits as earlier. Okay, again we obtain the same code as before. Okay, the wait for code. Of course, but now we will not send this. Instead, from the orange green cells, okay, what we will do is to pick B in these rows generated by the green rows, okay, such that if you look at the imbalance resulting for its sum, C plus B, this imbalance is actually minimized. And in this case, okay, this word over here will actually do the trick. And in fact, it does, okay? By adding these two words, you actually obtain this word which has weight 8 and is balanced. And some of you might notice, okay, this is the exact same example that I presented earlier with the extended Hamming code. Okay, so what it means that our previous observation is still correct, okay? Encoding and decoding complexity of this new scheme, okay, remains as big O of n log n. Okay, however, we must be a bit careful here. You need to come up with a clever way to find the balancing word, or rather balancing vector efficiently. Okay, so our game plan is this. Okay, we'll pick some balancing set B that's spanned by some rows of the polar matrix. Look at some space X. Okay, and what we want to determine is this quantity here, which is mu of XB, which I'm going to call the guaranteed imbalance. Okay, so intuitively, for some X, for some C, okay, we're going to ask how small we can make the imbalance be by adding factors from B, from the set B here. Okay, so we want to minimize this quantity here. Of course, then we consider over all possible C's, okay, and look at the worst case scenario. Okay, that's why we look at the maximum over here. Okay, so this is why we call this quantity the guaranteed imbalance. And so what we do in this work is to try to derive upper bounds on the guaranteed imbalance, okay, for explicit choices of X and B. Okay, so no random choice here. Okay, so just to be clear, okay, let me highlight some differences, okay, with the previous scheme by Mazumdar et al. Okay, so first, for DNA database storage, okay, B need not be a strict balancing set. So it suffices to have small GC imbalance. So what we want to do is to find a balancing set whereby mu of B, the guaranteed imbalance, is little O of n. Okay, i.e. the proportion of imbalance goes towards 0. Next, okay, X need not be the whole space. Okay, need not consist of all binary words of length n. Okay, this is because most of the time what we are doing is just simply to balance words that are polar cowords. However, this will actually lead to some chicken egg problem. So what we do is try to look at X, which is large, okay, so i.e. close to the entire space, so that it's likely to include a polar code. Okay, finally, and more critically, okay, we need B to be spanned by rows of GM, okay, the polar matrix. Okay, this is important so that we can apply the successive cancellation decoding algorithm, okay, of the polar codes. Of course, we will try to choose as little rows as possible so that the rows are, and also choose rows that are unlikely to be frozen. Okay, so for the remaining of this talk, okay, I'll try to look at a very specific set of balancing set BM and try to look at its guaranteed imbalances. I also try to roughly sketch how to lower the imbalances for short block lengths by constraining the space X. Okay, so what is this BM here? Okay, so BM refers to the span of the rows with half the dimension of the polar matrix. Okay, so let's be a bit concrete here. So M here is 3, and now I chose M here is 3, so therefore the dimension is 8. So we are looking at the rows with weight 4, and here these are the rows of weight 4, and I highlight them in green. Okay, so what we show is that imbalance, okay, guaranteed imbalance of this balancing set is square root of n. Okay, so let me make some little remarks. So first, okay, this is a property of the polar matrix. Okay, so basically for BM, the size is M. Okay, in other words, okay, the BM is always a span of 1, 2, 3, or sorry, a span of M rows. So in this case, we have 3 rows, okay, which is simply log n. Now, what we have essentially shown is that mu of the imbalance, okay, is little O of n. So this means that the proportion of imbalance goes to 0 as n goes to infinity. And what we notice here, okay, all this while as we let n goes to infinity, what the rate penalty that we pay is simply just log n. So therefore, this is actually negligible, okay, in terms of rates. Finally, okay, I think for many coding theories out there, okay, you will notice that the rows I've chosen is actually closely related to the famous Reed-Muller codes, okay? And indeed, if I include this last row over here, okay, the green rows and this last row, the all one vector, okay, this will actually be the first order Reed-Muller codes. Now, in the interest of time again, I will not derive the guaranteed imbalance. Instead, I will simply demonstrate how to obtain the balancing vector by a simple example. And a key ingredient in this step, okay, is this famous matrix here known as the Hadamard matrix. So again, the Hadamard matrix is also another tensor product, and the tensor product of this plus minus 1 matrix. Okay, so here again m is 3, so we obtain this 8 by 8 matrix of dimension 8. And so suppose if you want to balance this word x, which is 0, 0, 0, 0, 1, 0, 1, okay, what do we do? Well, what we're going to do is to transform it to a plus minus 1 vector by turning all the zeros to 1 and the ones to be minus 1. Okay, what we do next is to multiply the Hadamard matrix with U. Okay, then you obtain this integer value vector. Okay, 4, 0, 4, 4, 0, 0, 4, 4, 0, 0. Okay, well, turns out that these integer vectors here, okay, actually corresponds to the imbalances when you obtain by adding vectors in B. Okay, so let me convince you via example. Okay, since you are interested in the smallest imbalance, okay, we look at the value 0 here. And then we look at this corresponding row here, which is the third row. Again, this is in plus minus 1. So we can convert it back to the 0, 1 domain, okay, by turning 1s into 0s and minus 1 into 1s. So this is the B that comes up from it. Okay, and it turns out this B is actually belonging to our balancing set over here. And if you believe me, and if you want, let's check. Okay, if we add x and b, what do we get? If we add x and b, we check that the resulting vector is 0, 0, 1, 1, 0, 1, 1, 0, and its imbalance is 0, okay, as predicted over here. Okay. All right, and so what we have is a balanced word. Okay. Well, what is not obvious is that can actually show that this balancing vector can be found in this whole procedure that I described earlier. Will yield the balancing vector in big O of n naught n time. Okay, again, I glossed over many details, but some of you might find this whole procedure oddly familiar. Okay, basically, it's actually the vector in red that I had earlier, or rather the vector of imbalances, is more famously known as the Welch spectrum. Okay, and what I've done here essentially can be used to obtain the classical and non-classical proof of the covering radius of the first order Reed-Muller codes, okay, which is around square root of n also. Okay, so what we have shown is that if I give you this matrix here, the GM here, and look at this green rows, okay, this set BM, the guaranteed imbalance is at most 4. Also here M is 4, huh? So square root of 16 is 4. Okay. So what does this mean? This means that if you fill in anything on the orange cells here, okay, I can always find a vector B in this green rows, okay, in the span of this green rows such that this imbalance is almost 4. Now, can we do better? So it turns out the answer is yes. What we can do is try to, instead of allowing me to fill in anything on the orange cells, okay, we just pick away two of the top cells, okay, so freeze the first cell to 0 and the second cell to 1. Turns out if I do this trick here by sacrificing 2 more bits, okay, turns out that I can achieve perfect imbalance. Okay, in other words, no matter what you fill in the orange cells, okay, I always can find something in the green cells such that the sum is balanced. Okay, that's what this guaranteed imbalance means. Okay, and so in our paper, in our conference paper, we actually played with this idea, and by freezing various combinations of the bits, and here is a summary of the imbalances that I achieved. So basically, for what you can see, for n up to 128, or rather m up to 7, okay, we actually can achieve imbalance of 0, okay, by doing the suitable adjustments. Okay, in our conference paper, okay, we also look at using other balancing sets, okay, which are essentially higher order rate Miller codes, okay, and we try to give an upper bound on the parenting imbalance. And this work is really at a very rudimentary stage, and we find that there are still many things to explore. But essentially, the fundamental problem is this, okay? We have this quantity, the guaranteed imbalance, which is determined by two parameters, okay? X, the space, and B, the balancing set, okay? And we have a trade-off. So on one hand, we want X to be big. On the other hand, we want B and also the value U to be small. And so the fundamental question we are looking at is if we fix two of the three quantities over here, okay, how big or how small can the third quantity be? Okay, and this unfortunately is still largely unresolved. Okay, good. Okay, so that I have come to the end of my talk. Am I doing good in time? Okay, what happened in time? Okay. Am I going? Okay, cool. Okay, so I'm still good on time. Okay, good. All right. So this brings me to my conclusion. Okay, so in summary, what I've done in this talk was to look at combining balance codes with other features, and the central theme of this talk was this Knuth balancing technique. So what I did was to adapt Knuth's balancing technique to in a variety of settings, okay, peculiar to DNA-based data storage. And in all scenarios, okay, we have retained, okay, the efficiency and simplicity of the original balancing method. Now, to be very, very clear, okay, there are many other works in the community that look at similar issues. Okay, so there are work on GC balance, and with additional constraint, this additional constraint called the homopolymer run constraint. Okay, so basically you constrain the runs of the same symbols. What else? Okay, also there's another huge community, okay, working on this issue related to DNA-based data storage, okay, whereby they try to address the unordered nature of DNA sequences, okay, this notion of coding over sets, and also this notion of shuffling channels. Okay, I apologize for not mentioning these works during this talk. Okay. Also, there is also another group of work, okay, related to DNA-based data storage that I'm sort of involved in, at least part of it. Okay, this revolves around the theme of string reconstruction, and I don't want to go much into detail, but basically, Ogisa gave a very nice plenary talk during this recent ISIT, and I believe the video is still online. Okay, and so if you're interested, okay, here is the link to the address, and here's the QR code. Okay, so yes, so this is Ogisa. She was my postdoctoral supervisor at Urbana-Champaign, and I'm actually quite very grateful for her for introducing me into this area. And Ogisa is really very active, okay, for those who know her. Yeah, and she has been leading this thing on DNA-based data storage at both the theoretical and the experimental front. Okay, so if you're interested, okay, on a different group of works that revolve around DNA-based data storage, okay, feel free to check out her talk. What else? Okay, so finally, okay, I also mentioned during my talk that during the recent ISIT 2020, there has been a number of talks on DNA-based data storage. And I also personally basically compiled the relevant talks on my personal website, okay, and you can find this link on my on this page over here. And here's a QR code to my to the site. Yeah. So for those who are aware, okay, during this the recent ISIT, it was virtual, okay, and so we all presented our talks on videos, and I believe they are still available online. Okay, so if you're interested, feel free to take a look. Okay, so this is my final slide, okay. Okay, and I'm happy to take any questions.
Thank you so much, Han.
Yes.
So your
Thank you,
last
Professor.
slide has a QR code and there is a DNA strand in between it.
Yes, yes.
Is that part of the code or?
that is an error, which you can correct.
Right. So I had a quick question about
Sure.
this while like maybe as we are waiting for some other questions, perhaps. So about this basic problem that you showed at the end about this balancing thing, right? So you had three quantities U, X, and B.
Yes. Yes.
So are there any polynomial based approaches to this problem? Like,
Sorry? What do you mean by
are there
polynomial?
like you can view these codes as also, like for instance, the Reed-Muller code. So there's a polynomial description to the code. So
Oh, that will be interesting. Yeah, that will be interesting. So yes. In our ISFT paper, we did not use the polynomial description to get upper bound on the imbalance. Instead, what we did was to look at its geometry. So we used the affine geometry representation. Basically, numeric codes are uh, the minimum weight words are actually affine subspaces of a certain codes. Okay, and we use that to upper bound the imbalance. We have not tried to use polynomial representation. Okay, it might work. Okay, but it wasn't very obvious to us. At least how to adapt it to guarantee the imbalance. Yeah. So it might work. It might work. Yeah, just that we couldn't see how to do it.
okay. So there is a question. Yeah. So Lalita, you want
How
to
come I
Please
don't
go
see the
ahead.
questions? Yeah.
So there is a question from Krishna Gopal.
Oh, I can't see anything.
It is okay. So I can just read it out. Perhaps you can answer.
Okay, sure.
So you have mentioned a term trace reconstruction in your slide. Can you please say
Yeah.
something about it? That is a question.
Yeah. So this was some works that was presented by Ogisa. Okay, if you are interested, yeah, you can look at her talk here. Okay, yeah. So let me just roughly tell you what it's about. Okay, so I have to be a bit more careful here. This is coded trace reconstruction. So the original trace reconstruction problem is this. Okay, so I give a sequence here, original sequence, and you sample it multiple times. Okay, each time there will be a probability of certain bases being deleted. Okay, yeah. So original trace construction, you're just looking at deletion. Okay, so you get multiple copies of a sequence. Okay, or of one sequence, and your question is, okay, given a certain number of traces, are you able to reconstruct the original string? Yes. So in Ogisa's work, okay, they look at a variance because so basically for data storage, okay, we can control design what the sequence is. So you look at the problem whereby your sequence is not really any arbitrary sequence, but rather a coded sequence. So whereby you have certain information on it. Yeah. So in there, I think this I think it's on in transactions now. Yeah. So in the recent transactions work, they actually look at this problem, and yeah, in addition to just to code it such that you can reconstruct with little traces, he also introduced this GC balance notion, okay, to again well to decrease the probability of deletions, for the physical channel. Yeah. Okay, I hope it makes sense now.
Okay.
Have I answered his question? Yeah.
Okay. So I think you have, because he's not. He doesn't have a follow up. Okay. So any other questions?
Okay, I see now. Oh, I see, I can see. Oh, I can see the question. Okay. Okay. Oh, cool. Okay, sorry. Now I found it. Okay. Okay. Sorry. Uh-huh. Okay. Oh, thanks, Wang. Okay. 20. Okay. Hi, Hong. Can you see this? Okay. Do the constructions avoid secondary structures? Yes, then how? Okay, when was this asked? Let's see. Sorry, I see a question. Should I answer them or?
Yeah, you can, but maybe this like technical questions we can take it immediately. So
Okay. Okay.
so Lakshmi, you want to go ahead?
I guess let answer the questions in this box, Q&A box.
Okay.
I'll wait.
Sure.
Sorry.
No, you can just talk, right? So I guess you can just.
Okay, I can just talk, right? Yeah. So okay, let me answer. Let's go and okay. All six. So Krishna asked about the avoiding secondary structure. If yes, then how? Okay. So I believe you're looking at the primer, the very first part of my talk, whereby I talk about primer dimers. Yes. So I can't remember the details. Okay, but yes, we can modify our techniques. Okay, to avoid secondary structure. I can't remember the details, but yeah. Anyway, the details are in the paper, so if you're interested, you can look it up. Yeah, but again, although we avoid secondary structures, I think there was some constraints on how long these structures must be. Yeah, because if it's longer, then it's easier to avoid. Yeah. So yes, that's what I roughly remember. Okay. Yes. Okay, so there's this Mohammed, is it? Mohammed?
Yeah. So he's asking a question.
How
So can you see his question?
how it will be good to pursue full stop in this area? Works in competition. Okay. I I I don't know how to answer this. So are you asking for a position? Yeah. Unfortunately, this is 2020 and some things have changed due to you know what. Yeah. So as much as I would like to hire, things have changed quite drastically in the political climate in Singapore. So I cannot give you any answer. I'm not sure how's the hiring process now. How's the hiring market now in the States also. Yeah. Everyone is stuck in their own bubble. So I'm so sorry. I cannot give any advice now. Yeah. Things have changed so much in 2020. Yeah, I won't imagine me giving a talk like this time last year, or even this time like 4 months ago. Yeah, so okay, so sorry. Yeah, so Mohammed, I can't answer your question. Yeah, so sorry.
He says there are very few people in this area, so I'm worried about it. So I guess you can give some generic comments if you can. So how to go about
Yeah, it used to be. Yeah, I used to think also that there are very few people in the area, but the recent FAT proved me wrong. So that yeah, so I know of groups in Israel. From even in Singapore, we have a group. Yeah, we have at least two groups. Where else? Where else? Israel, the States, of course. Yeah. Uh, yeah. I really don't know how to answer this. So sorry. Yeah. Yeah. Mm, mm.
Okay. So I think Lakshmi. So you want to go ahead?
Okay.
Yeah. Hi, Anne. My name is Lakshmi. Thank you very much for the talk. So I have one question related to the third part of the talk. In one particular slide for Poda codes, you had chosen non-zero frozen values for some of the rows.
Yes.
My So my comment is that you probably chose it because now you make the code effectively non-linear. I'm not sure if that is helping you in some way. And my question is there a systematic way to see which bits have to be set to 1 among the frozen bits?
Okay. So okay. So yeah, good question. Okay. Okay. So let's come back. So I think you're asking me why I freeze this to 1, right? Okay. Okay. The first freezing of 0, I think it's a bit obvious. Is that we are trying to make sure that all the words have even weight. Yeah. So this is typical. Yeah. So if you work in polar codes, this is a very typical observation. Okay. Yeah. So I'm freezing this to 1. Yeah, partly yes. Is to make it non-linear, but actually, it was really what we observed in our what do you call it? In my my proof, which was that. Oh, sorry, I think I freeze the wrong row. I think it should be this one. There's 1. Okay, so sorry. Yeah, wait. Because oh no, what is this? This very good. This one wrong. Oh, okay. Yeah, it's supposed to be this one to be 1. Oh, shucks. I made an error. Anyway, yeah. Okay. So what I'm trying, I'm blabbering. Okay. Okay. Good. Okay. As to why I froze it to 1. Okay. Again, you have to look at the proof. Okay, it's quite a combinatorial proof. That yeah, I made use of the Hadamard, the some characteristic of the Hadamard matrix. Okay, to see that yeah. I try to think of a way to explain without going too much into too much. So, okay. So again, it's a manifestation of the proof. Okay, as to why I decided to freeze this to 1 instead of 0. As to whether there is a systematic way, I would really want to find out. Okay, but as I said, this was only a beginning of our investigation, so we couldn't have. We wanted to find a systematic way, so we couldn't find it yet. Yeah, so it would be nice if someone could come up with a systematic way of finding this freezing pattern. Yeah. Okay. Yeah. So yeah, this is still very open. Yeah. We just
Thank you.
Follow-up question. I just follow-up question.
Sure.
Did you have did you had lower
Sorry,
right?
sorry?
As a function of you had a upper bound on the guaranteed imbalance of your scheme?
Yes, sure.
Okay.
Okay. Do I have a lower bound, right?
Yes.
Okay, so the question is whether do I have a lower bound?
Or upper bound is also okay, yeah.
So what do I mean by lower bound? How do I click? Maybe I did. Okay. Okay, so this one, right? The lower bound on the imbalance. Hey, why is this? Sorry. I'm trying to move this such that I can see. Okay. Do I have a lower bound on this thing? Yeah, it doesn't. Okay. Okay, sure. Good night, home. Just say good night. Oh. Okay. Oh, Navin, good question. Okay. Other than cyclic codes, are there any way of using the automorphism groups for balancing purposes? Okay. Hi, Navin. That is a very good question. I don't know why I can't answer. Wait, anyway. Okay, hi Navin. So your question is, other than cyclic codes, is there any other way of using the automorphism groups for balancing purposes? I think the answer is yes, but I haven't really looked at it. Yeah, we haven't really looked at it. I would suspect it will work. Just that we haven't really tried it out. Yeah. Yes, you're asking a question which I have sort of want to try to explore now, but you suggested it. Yes, so I haven't looked at it yet. Yeah. Yes. Yeah, this is an interesting question. Okay, yeah. If we look at other automorphism group, we have to make sure that one obstacle that I see is that if we use another automorphism group, we may not be able to guarantee that there is one of these representatives that actually is balanced. Yeah, so the cyclic shifts is nice in the sense that when I move it, I can see that one bit goes in and one bit goes out. And so in some way you can have like what Knuth did, okay, to ensure that the changes in weight is always missing by one. If we use another automorphism group, yeah, we have to be careful about doing that. Yeah, to make sure that yeah, the change in weights is always differs by one. Yeah. I hope I answered your question. Yeah.
So this may be a related question. So this log n redundancy which you have for your balance construction, right? So is that also the lower bound for the redundancy?
No. Okay. The lower bound for the redundancy is half log n.
Okay.
Yeah. So yes. So there's a interesting line of work, okay, by Iming and Weber, whereby they try to improve Knuth's original construction, which uses log n bits of redundancy to half log n redundancy. Yeah. And this proof is basically quite easy. You just basically look at how log of 2n n choose n over 2 behaves. Yeah.
Right.
You realize, yeah, you have a half log n. Okay. Yeah, but I think as of now, I'm not aware of any construction which is of fixed length, which achieves a half log n. Yeah. Okay, hope it answers your questions. Oh, good.
So this is like one more generic question, perhaps. So when I look at this general area of this